Yes, I hope you're good from your end. So this is Kenya National Examination Council, Kenya Certificate of Secondary Education, KCSE, Biology Theory, Paper 2, 2018. So this is the KCSE paper that was set back then in the year 2018. We are here for the revision purposes for the Form 4s who are preparing for their exams, national exams. So this is how your paper will look like. Biology paper 2. That is the cover page with instructions. Let's dive to the questions. That is the business of the day. Section A, 40 marks. How are these 40 marks coming? We are going to see. As for now, I'm only going to talk about question 1 and question 2. Next paper or next day, we will dive more on the question 3 and 4 until we finish this 2018 paper 2 biology. So stay tuned. Kindly revise these papers. They are very, very important because some of these questions are being repeated. So number 1, the diagram below illustrates a section of mammalian kidney. So we know that mammals have kidneys, and including you have a kidney. What is the role of kidney? Is to filter blood. They filter blood to remove what? To remove wastes or to eliminate waste. That is the role of the kidney. So let's dive. So we have a mammalian kidney down here, a cross section. So we have region G and region H. Here we have the arterial. There are two. One, afferent, that is wider. Next, we have efferent. This one is narrow as compared to efferent. So there is a reason. And then from here, we can easily see that E is the glomerulus. There is the Bowman's capsule that surrounds or it encloses the glomerulus. And then this kind of part here is referred to as, these are blood vessels. So it is just a blood vessel. This is just a blood vessel. Or they can be called branch of renal artery. It is a branch of renal artery. You can see it is a branch of renal artery. So being that the blood enters the glomerulus, it enters with high pressure inside the glomerulus. So remember, afferent arterial is the one that is wide as compared to efferent arterial. Why is it wider compared to efferent arterial? To create a high pressure resulting to ultrafiltration. So that is what is going to take place at this region G first, ultrafiltration. That is the first process that will happen at this point G, just right within the glomerulus here. So what happens within the glomerulus is ultrafiltration. So the blood enters via higher pressure. And then ultrafiltration is going to be done what? To take place. Another thing that is going to happen now, we have now right here the proximal convoluted tubule, and right here we have the distal convoluted tubule. So these tubules, what do? They do what is called selective reabsorption. Selective reabsorption of glucose is one of them. Another one is vitamin and even excess water. that needs to be reabsorbed. So that is what is going to take place at the proximal convoluted tubule and distal convoluted tubule. So those two things are going to happen in region G. And then down here, we have the loop of Henle. This one is called descending limb or arm of loop of Henle. And then you can easily see that it descends and then ascends. So the ascending one, they plays a role. The descending one plays a role of selective reabsorption of water. That is what the descending loop of Henle does. Ascending does what is called active reabsorption of salt, of sodium salt. Sodium what? Sodium salt. So get the two differences. So what happens in region G? In region H are now selective reabsorption. One in the distal or at the distal, at the descending limb of loop of Henle, what happens there is selective reabsorption of water only. And then at the ascending loop of Henle or ascending limb of loop of Henle, what happens is the reactive reabsorption of sodium salts. So those are generally what is going to happen within this mammalian kidney. And then we have the collecting duct here at this region of H, still collecting duct. This collecting duct, what does it do? This one, this is the duct I'm talking about, this hollow part. What it does, it also does what is called selective reabsorption of water. So in general, we have talked about the entire diagram. So let's dive to the questions. Name the structures labeled E and F. E, we have talked, it is glomerulus. Remember, biology spellings are very, very important. Which part F is that? We've talked about it, blood vessel. Blood vessel or branch of renal artery. It can be called blood vessel or branch of renal artery. Are you getting that? Yes. So those are the parts. Question B, explain the process that take place in regions labeled G and H. I have already explained everything. I've said that this region labeled G, what happens within at the glomerulus? What happens there is ultrafiltration. At the proximal and distal convoluted tubule, what happens there is selective reabsorption of substances. So let's dive. So we can say that at the glomerulus, the afferent arteriole is wider compared to the efferent arteriole. This creates high pressure. This creates high pressure, resulting or leading to ultrafiltration. Yes, so that is what is going to happen. or the process that is going to take place at that region, G. Just like I said, it is at the glomerulus. First activity or first thing at the glomerulus. What is going to happen is that at efferent arteriole, being that it is wider, there's a statement that has been left. So it is wider compared to. For that high pressure to be brought, what happens? It is not being wider. There is a statement I have left. We need to jot it down. So it is wider to bring blood to the glomerulus. Are you getting that? It is wider compared to arterial, efferent arterial, as it brings blood to the glomerulus. So we need to jot that down. So that is a very, very critical point. So we can say that efferent arterial is wider compared to, afferent is compared to efferent arterial to bring blood, to the glomerulus, full stop. So is when we say this creates, so these two didn't erase themselves here, huh? Okay, I will deal with them properly. So this creates a high pressure, resulting to ultrafiltration. Full stop. So we have known that at the glomerulus, the efferent arteriole brings blood to the glomerulus, and it is wider compared to efferent arteriole. That action creates higher pressure, resulting to ultrafiltration. Another thing is that at the proximal convoluted tubule, at the proximal convoluted tubule, and diastole convoluted tubule, the substances needed by the body that is glucose, vitamins, and excess water. So at the proximal convoluted tubule and distal convoluted tubule, the substances needed by the body, glucose, vitamin, and excess water, are selectively reabsorbed. Selectively reabsorbed. There is where now they begin to be reabsorbed. Are you getting that? So two activities happens there: ultrafiltration and reabsorption. So that qualifies you the 3 marks. Region H, we have said that at the descending limb or arm of loop of Henle, remember it is always capital H, at the descending limb or arm of the loop of Henle, selective reabsorption of water takes place, full stop, at the ascending limb. It is limb, not limb. Limb. Or arm of loop of handle, comma. What happens there? Sodium salts, in bracket Na. Sodium ion salts or sodium salts are actively reabsorbed. Are actively reabsorbed. Full stop. Again, at the collecting duct, comma, water is selectively reabsorbed. Full stop. So those are the three activities that happens or the processes that takes place at those particular points. And that earns you how many marks? 6 marks for free. Question 2. I said I'm going to do question 1. We have talked about the mammalian kidney, and we are now diving to the next question. How many marks have we garnered in question 1? 3 plus 3, that is 6. Plus 2, 8. Remember, paper 2 biology. 5 questions are 8 marks each. 8 marks each, totaling to 40. And that is why you saw 40 marks here. Section A. 5 questions. Section B entails the graph part. The graph part is 20 marks plus 40, that is 60. And then essays now come from question 7 or 8. So you choose either 7 or 8. So the structure is simple. The first 5 questions, most of them are, there must be some diagrams among them. If I'm not wrong, there must be diagram. And in fact, most of the cases, those questions are diagram based. And they are structured questions. Very simple. Once you know what takes place, you are good to go. And these questions always come from the topics, e.g., nutrition. Number 2, excretion. Like this one, number 1 is from excretion as the topic. Another one is genetics, gaseous exchange, respiration. Can you jot them down? Because those are some of the topics that are very necessary for paper 2. Response, reception, and even coordination must come. The bones must come. So you need to know such topics. And lastly, I will talk about meiosis and mitosis. I think I've talked about genetics. Genetics also is always there. They always ask genetics and they always set it. Very important. The crossing, the diagram, the cross diagram is always being set. So those are some of the topics that you need to know as a learner. So let's dive to question 2 and see which topic is this one from. Below is a graphical representation of students' height in a classroom. So we have a graph here. Let us see. So the number of students are increasing. They are from 2 to 20. And then we have what here down? We have height. Height is in centimeters. So it therefore means that the height increases from 120 up to 189. 120 up to 189. So this kind of a graph here that we are seeing here, this is just normal distribution curve. Normal distribution curve. It tells us how the height of learners increases up to the maximum or optimum, which is 160. Which is 160. You can see from the diagram. And then after some times, the height begins to decline. So it therefore means that few learners are from 120 to 139, and then you see how the height increases. So not defined. We cannot define that the height is of maybe the number of students is just constant 120. No, there is a range. There is a range from 120 to 189. Some are 166, some are 189, some, I mean, the others that are 130. So like when you come here, 16 learners have the age of 160. Just imagine, 16. Others, 6, they have around 142. So it is not constant. It is not defined. Are you getting that? Where we have a range, we know what is that. So let's go. Name the type of curve illustrated. I have said it is the normal distribution curve. Normal distribution curve. State the type of variation represented by the curve. So when you hear about variation, which topic is that? Genetics. That topic is genetics. So just like I was mentioning some of the few topics, the likes of gases exchange, the likes of nutrition, genetics, reception, response, and coordination. Those topics must always be there. Respiration, huh? They will be there. Gases exchange, they will be there. So kindly, if you want to pass biology paper, it's very simple. Graph, know how to plot the graphs. 20 marks, how to answer those questions. In fact, answering those questions are very simple, and you will see. I will tackle that question. It is very simple. Plotting and then answering. 20 marks for free. And then from there, we have an essay. You write whatever you know concerning whatever you've been asked, and let it be true. You know, biology is very, very true, and it is exact. We don't guess here. We speak of facts. Factual responses are needed, particularly to paper 1. Factual responses. So this is a normal distribution curve. State the type of variation. So we have two types of variation. And what is variation? Variation are just observable differences among different organisms. So those differences. Now you realize that there's a big difference here. There are others that have 189 in terms of height. Other students have 120. So there is a difference. We can easily see the height in terms of tallness and shortness of the number of students. And we can tell that this particular student is this tall, is this short. So that is what we are referring to as what it is called observable differences. Are you seeing that? Now, when you talk about height, that is a continuous variation. See how the curve is continuing. Right from 120, the height increases up to optimum, and then it continues to decline again. So that is continuous variation. There is no defined range, but rather there is a wide or there is a range. Are you getting that? That is continuous. So continuous variation. So there are two types of variations. Number one, continuous, just like I've said. It is continuous from the graph. Number two, there is discontinuous type of variation. In discontinuous type of variation, the range is defined. It's a very defined type of variation where you can easily tell that this person is male or female, and that is sex. So that is discontinuous variation. Example of discontinuous variation, one of them is sex. You can easily tell that this person is a male and this person is a female. And then another example is ability to roll the tongue. Ability to roll, roll is double L. ability to roll the tongue. If you don't know, you don't know. If you know, you know. That is a defined activity. Are you seeing that? So in discontinuous, it is a defined type of variation. Continuous, it is not a defined continuous variation. There is a range. There is a continuous variation among different organisms. Are you getting that? Yes. So those are the two differences. State two meiotic processes that lead to variation among organisms. So one of the meiotic processes is crossing over. Crossing over. Number two is independent assortment. So remember how chromosomes pair, homologous chromosomes, they pair together. That is what we call crossing over. And when they pair, when the homologous chromosomes pair, a process called synapsis, when they pair, the variation, the chromosomes exchanges their genetic materials. Are you getting that? The chromosomes, they are carrying the genetic material. They're the ones carrying the genetic characteristic, the information about such organisms. So when crossing over is taking place and there is pairing of homologous chromosomes, what happens is that variation is going to be done. It's going to be brought about. Variation is going to be brought about. That differences I was talking about will be brought about when crossing over has happened. Independent assortment, in this case, you realize that when chromosomes, again, they pair unknowingly, the chromatids, they pair unknowingly. So there is no that non-pairing. They pair by default. When that happens, they sort themselves independently, unknowingly. Nobody is telling them, you need now to pair with this one. You need now to pair with this one. When that happens, there is a variation. There is a variation. There must be a difference. Are you getting that? So those are the two meiotic processes that leads. Now they happen within meiosis 1 and prophase 1 each. Not each, but different. Explain the role of variation in organisms. Explain the role of variation in organisms. So what does variation bring in organisms? So number one, variation leads to organisms with leads to production of organisms, leads to rise or production of organisms. Hey, this my pen is slow in erasing. So let's give it time. Finally, variation leads to rise of organisms with advantageous traits. It leads to the rise of organisms with advantageous traits that are selective and are adaptive by nature. They are adapted to nature. So there are those organisms that are naturally selective. They select the environment and then they adapt to that particular environment. And then all of a sudden, they give rise to those traits that are of advantage. Are you getting that? There is propagation of new offsprings or propagation of young ones with advantageous trait. Are you getting that? So that is what variation can cause. It can lead to rise of organisms with advantageous traits that are selective and are adapted by nature. Are you getting that? And then variation again can cause propagation of desirable traits. Are you getting that? So it can also cause propagation. Once these advantageous, hey, what did I write here? Advantageous traits. Advantageous traits. Advantageous should be A. Advantageous traits. So once these organisms with advantageous traits have been naturally selected and they have adapted to a certain ecological niche or environment, what happens is that now they will propagate, multiply. The desirable trait will multiply. Are you getting that? So the organism will mate and then a lot of production of the desirable traits will happen. That is what we call propagation of desirable traits. So that is what variation can cause. It can cause propagation of desirable traits for future generation. So those are the two roles of organisms, variation in organisms. Last question. I said I'm only going to do question 1 and question 2. The third question or question 5 will be done, not question 5, but question 3. Should be question 3, yeah? Should be question 3. I will do it the following day. Or if you want me to do it, let me just do it. No problem. I will do it for the sake of you as a a Form 4 learner who is preparing for the KCSE. Let's dive to the last question of this number 2. Explain the need for genetic counseling in the present day health facilities. Very nice. Genetic counseling, genetics. So those who does genetic counseling are called genetic counselors, or they can be geneticists. Are you getting that? So what are their role? Number 1, they provide need for genetic counseling, not they now, it provides. It provides advices on the risks. Provides advices on the risks of. Provides advices on the risks of genetic disorders. Genetic disorders. So remember, these genetic disorders, one of them is sickle cell anemia. One of the diseases brought by genetic disorder. Genetic disorder, it means now the genes are not in order. Are you getting that? The likes of now, like I was talking about you having maybe a lady having lacking the X chromosome or having one X chromosome, a man having YY chromosome. Are you getting that? Or a lady having XY chromosome. That is the genetic disorder I'm talking about. Are you getting that? So when the genes are not aligned the way they should be, it creates a genetic disorder. So provides advices on the risk of genetic disorders, e.g., uh, risks of sickle cell anemia. Now, genetic disorder, another genetic disorder, we have cystic fibrosis. That is another genetic disorder. Huntington's syndrome. Those are some of the genetic disorders we may experience. Are you getting that? Yes. Number 2, role of or why we need genetic counseling. Why we need genetic counseling. Now, it provides support services to families. It provides support services to families. E.g., what are these support services? Refers to a health professionals. So one of the support services is referring the families to a health professionals, to health professionals, to health professionals. So that is another role, another importance why we need to go for this genetic counseling. Because if we are not going for this genetic counseling, where will we get these health professionals? Are you getting that? So that support service to families is brought by us going for genetic counseling. Are you getting that? So again, it helps identify or know the genetic disorders. So instead of even providing advices, we are going to identify and know these genetic disorders, e.g., sickle cell. We have cystic fibrosis. We have Huntington's disease, etc. There are so many. There are so many. So those are some of the need why we need to go for genetic counseling. So I'm just going to dive to a few questions under this question 3, because too much of something is dangerous. The photograph, tomorrow is also a day. Photograph below represent a plant in certain division. So there is a plant here with different parts. So let's see. This part here is called the sporangium. This part is the seta. Not stem, seta. This part, these are the rhizoids. So this particular plant belongs to a division Bryophyta. They are Bryophyta. They have rhizoids, so they don't have the true leaf, stem, or rather the roots that we know. So they are Bryophyta. They reproduce by use of spores. So that is asexual reproduction. Asexual reproduction called sporulation. Called sporulation, where now the mature sporangium and the sporangium, the role of sporangium actually is to protect the spores. Are you getting that? They surrounds the spores. So that is sporangium. So spores are here. What happens is that once the spores are matured, it bursts. The capsule bursts, releasing the spores into the environment. And then upon receiving the favorable factors within the environment, the spores grows back to the parent plant. So that is sporulation. So let's dive to the question. Name the division to which the plant belongs. I have said division is Bryophyta. With reference to the photograph, state 3 observable features of the division named in one above. Observable features. Number 1 is the hair-like rhizoids, or we can talk about numerous rhizoids to provide anchorage and absorption of water and mineral salts. That is the number one observable feature. Number two, we can talk about this the sporangium. Which supports the spores. The last one, we talk about the seta. In fact, seta is what provides support to the sporangium. Provides support. The sporangium does not support the spores, but surrounds the spores or protect the spores. Protect the spores. Still, seta now supports the sporangium. So those are some of the three observable features. Name the parts labeled N and P. N. N, we have talked about sporangium. P is the seta. This is the seta. Explain how the part labeled Q is adapted to its function. Part labeled Q. Wow, the rhizoids. Rhizoids, so we can talk about they are numerous. They are numerous to provide a large surface area for maximum absorption of water and mineral salts. Number two, they are numerous still. There should be an O here. So numerous. They are numerous to provide anchorage. They are numerous to provide anchorage, or they are hair-like. So you can talk about, instead of saying numerous, they are hair-like. They are hair-like to provide anchorage. That is the observable feature. And that marks the end. Too much of something is dangerous. We will end from there. We will proceed next time. Thank you for listening to Teacher Zamalek. Kindly go through this paper. A very, very nice paper. So you need to know excretion as a topic in general, the kidney nephron, how it functions, and how the process takes place there. Number two, you need to know genetics in broad terms, broad details. Cross diagrams should be known. Today, there is a graph that has been brought, the normal distribution curve, and there is a continuous variation. And we have talked about meiosis, again, role of variation and the need for genetic counseling. So in general, you need to also go through genetics. If they will not bring the closed diagram question, they will bring these ones. Again, the next question is the topic here referred to as classification 2. So there is classification of this plant. We have the kingdom plantae. Kingdom plantae. Kingdom plantae has 3 divisions. And those divisions, I want to write them here down. The first division is pteridophyta. The second division is bryophyta. These pteridophyta, they include the likes of fern. Now, this fern has the reproductive parts known as sorus or sori found on the lower side of the leaf-like part of a fan. It is a leaf-like on the lower side of it. Are you getting that? So one of the example of a pteridophyta plant is fan. Bryophyta, one of them is the moss. Are you getting that? Another one is liverwort. So those are some of the divisions. The third one, spermatophyta. In spermatophyta is again divided into two. Number one division is angiosperms and gymnosperms. Gymnosperms, they have naked seeds. Angiosperms, they have enclosed seeds. One of the examples of gymnosperm is cedar. Cedar have got naked seeds. Angiosperm is again divided into monocots and dicots. Are you getting that? That is how these things happens. That is how classification in this kingdom plantae happens. And you have to know them by their characteristics. You have to know their features. In terms of division, you have to know their features. What they have that helps them. Are you getting that? Do they undergo alternation of generation? Do they have two reproductive the gametophytes and even the sporophyte? That is the alternation of generation. So they undergo two generations. So that is all about this paper as per now, 2018 paper 2 biology. That is the far I can go for today. Let's meet in the remaining questions. That is section B. We will talk more on section B. If there is a question that is lacking here, then we will again go and check on it and then we'll tackle it. Don't worry. Don't worry. Like I can see, we have just dived to question 5. It means that there are some few questions that are also missing in this particular paper. Number question 3 and even question 4 is not here. I will look for them. That is what I'm going to look for. And then I will come back with them. Kindly go through this paper. Tomorrow or next upload will be the paper tackling question 4 and question 3 and even question 6. Are you getting that? And then from there, I think I will dive to another paper because question 7 and 8, those are simple. Essays are very, very simple and meant for you. Bye-bye for now. Let's meet in the next paper. See you as you see me.